everyone, and welcome to Chapter 1, Part 2 of Cell Structure. We are here talking about micrometry, basically how do we measure cells, and also magnification. So let's start a little bit with just talking about units, okay? In the previous video, I mentioned that if it's a microscope, a light microscope that we're dealing with, we will usually express the actual size of objects in micrometer. The SI unit is meter. Suitable units for light microscope is micrometer. And in a rare case where they ask you to figure out the actual size of something under the electron microscope, that should be in nanometer. Now note, you should always measure image length in mm. Sometimes this is a requirement and there's a mark just for you to measure it in mm, not cm, right? So measure in mm, and then to convert it to micrometer, just times 1000 and present the actual diameter or length of whatever structure they want you to present in micrometer. So the problem also with measuring in cm is that usually people sometimes make mistakes and accidentally multiply by 1000 instead of, say, 10,000, right? So be very careful of how you are measuring and how you're converting. I recommend to definitely measure in mm and always times 1000 to convert to micrometers, because that is straightforward, and if it's a good habit, then you may never get it wrong. Anyways, let's go on to how do we measure cells. Now, before we go into anything, let me just show you the microscope. This is how it looks like. This is what we call the eyepiece. Eyepiece. This is what we call the lenses. This is what we call the stage. This is where the light is. These are some knobs are here, some knobs are down here, but in this case, these knobs here are the focus knobs, or that might be the hinge, actually, I'm not sure. But yeah, probably this or this, honestly. But the point is, you need to know two things. The eyepiece reticule is on the eyepiece lens. This is one thing we need to measure cells. The eyepiece reticule is here. Inside there is a lens, and printed on the lens is usually an eyepiece reticule. It's removable, so when people don't want it, they can take it out and put it back in when they need it. Anyways, the next thing we need is a stage micrometer, and as you guessed it, stage micrometer is placed on the stage. And it's because it's usually like a slide. This is how it looks like in the lab. It's usually they write the measurement on it. This is 0.1 mm, and they usually have like specific measurements on that slide. It's actually a really tiny ruler. It's a ruler. Oh no, what happened? Yeah, it's a mini ruler. Okay, and it gives you the actual measurements. The eyepiece reticule, these divisions are all relative. They are all arbitrary. And that's why we don't give it a unit. It's just arbitrary units. It's just relative. This here is usually in millimeter or micrometer. The mini ruler, after all. Now, what do we do with it? What do we do with it? Now, the stage micrometer, again, as I said, shows the true length, right? Usually, okay, it's 10 mm in length with 100 small divisions. But honestly, this depends on your labs and, what kind of stage micrometer they're using. Usually, though, it's 10 mm in length. So 1 cm, this whole thing here is 1 cm, 10 mm with 100 small divisions. So 10 divided by 100 means one small division is 10 mm divided by 100, which is 0.1 mm, which converted to micrometers is 100 micrometers per division. And you know what? When you zoom in using your lenses, so when you change from maybe a 10 times objective lens to a 40 times objective lens. By the way, objective lens are found here. These are objective lenses. You can rotate them to change to the power you want. And when you do that, it always appears bigger as the magnification increases. The trick question in past years is always, does the object increase in size? And the answer is no, it only looks bigger. It doesn't actually become bigger. So be careful with the English right there. Now, however, the next piece of equipment, the eyepiece reticule that we place at the eyepiece lens, because it's not under the objective lens, right? It's at the eyepiece, like where you are looking in. This scale would appear constant no matter the magnification. Usually it has 100 reticle units, or sometimes we just abbreviate it as 100 EPG, eyepiece reticule. These 100 divisions on the eyepiece reticule are arbitrary. It doesn't have a true value. The mm and micrometer is just a means of measuring it later. So if you want to use the eyepiece reticule, to measure stuff, we will need to calibrate it. This is kind of an example of how it may look. You can see that the eyepiece reticule appears constant in both images. However, the stage reticule, or also called the stage micrometer, appears larger when the magnification is increased. So again, the eyepiece reticule is constant, it's at the eyepiece, right? And the stage micrometer is a slide with a tiny mini ruler on it. And you cannot stack slides, it doesn't work that way, okay? So what we actually need to do is, number one, calibrate the eyepiece reticule with the stage micrometer. So find out one eyepiece reticule, right, which is relative, at the particular magnification, right, equals to how many divisions on the stage micrometer, and therefore how many micrometer would one eyepiece reticule be. And number two, then we can use this eyepiece reticule to then measure the cell and structure. So we remove the stage micrometer and we put in the specimen under the microscope. Then we look under it and use our eyepiece reticule to measure it. So it's a two-step process. I mean, you can switch it, it's fine. You can measure it using EPG and calibrate it, it's fine. It's the same, right? The point here is that we cannot use the stage micrometer to measure the cell or structure straight away on a microscope, because it is not possible to stack the two slides together and be able to view them both at the same time. So we need to calibrate the eyepiece reticule and then use it to measure. Okay, here's an example. Let's imagine this is you looking at a microscope. Your eyepiece reticule looks like this. It has 100 divisions, right? And it's at the eyepiece. The stage micrometer against the tiny ruler on the slide under your objective lens under the microscope. Now, what you do is actually look for two lines that line up. Okay, this is not a very neat line here, but imagine that they do line up, and figure out how many micrometers really is one EPG. So this tells me here is 100. Sorry, this tells me is in millimeters. So 0.1 millimeters is this length. 0.1 millimeters is 100 times 1,000, that's 100 micrometers. How many divisions does it line up with? How many divisions of the eyepiece reticule? That's 15 EPG here. Okay, be careful, because the smallest division, right, is considered as one eyepiece reticule. So how much is one EPG then? Easy. You just take 100 micrometers divided by 15, and you should get your answer, which is um 6.67, oops, 6.67 micrometers, rounded up to 2 decimal places. Now that we have this value, we can then go and take this stage micrometer off the stage, right? And then swap the slide with the mini ruler, stage micrometer, with your actual specimen. Then this is how it looks like. So imagine that this is how it looks like under the microscope. This is your specimen here, and you can see here that this is the eyepiece reticule. You have figured out that 1 EPG equals to 6.67 micrometers. Okay, so how long is this cell? So this is around 11. Okay, I'll just say 11 EPG. That's the closest guess, right? We don't want to try and guesstimate 0.1. Instead, just say 11. So that's just 11 times 6.67 micrometers. Sorry about that. Little bit glitchy. 6.67 micrometers. And with a quick calculator, that is around 73.37 micrometer, rounded up to 2 decimal places. That is the length of this cell, the actual length of this cell. Why don't you try another example? Okay, try this. Given that as 100 divisions of the stage micrometer measures 1 millimeter, calibrate the eyepiece reticule. The first thing you need to know again is to calibrate, right? There are 2 big things that you should think of here. Number 1, the stage micrometer could be magnified under the microscope, so you use that information to derive which is your eyepiece reticule and your stage micrometer. And hint number 2 is that, hey, 1 EPG, right? 1 EPG, 1 eyepiece reticule is the smallest unit. 10 EPGs, mark 1 here. So this is technically 10, 20, 30, 40, 50, because this tiny division, right, this tiny division is 1 division. Okay, you try. Pause this video, play it again when you're ready. Okay, have you tried it? Did you really pause? Pause it if you haven't. But if you had, let's look at this question together. So here, we know that the stage micrometer could be magnified under the microscope, but usually the eyepiece reticule is able to be seen throughout, right? So the bigger one here is usually the stage micrometer. And it seems to line up perfectly at this point and this point. Now let's look at the question. It says 100 divisions measures 1 mm. So 1 division, right, is 1 mm divided by 100, which is 0.01 mm, which equals to, right, mm to 1 micro is times 1000, so it's 10 micrometers. So from this entire distance, right, has 10 division. This is 1 division, right? So this division is 10 micrometers. Therefore, 10 divisions, right, on the stage micrometer. Okay, maybe I should have drawn it like here, because this is 10 micrometer. This is 10 times 10 micrometer, which is 100 micrometers. Let's look at how many EPGs that lines up with, right? You can see that in 100 micrometers, there is 10, 20, 40, 50 EPGs. So 50 EPG, right, 50 eyepiece reticule divisions equal to 100 micrometers, as this tiny ruler tells us. So what is 1 EPG? That's 100 divided by 50, which is 2 micrometers. That is how you calibrate this particular eyepiece reticule. Again, the smaller one is the eyepiece reticule. We know that because it's not magnified. The larger one, this diagram, is the stage Oh, micrometer. Um, so yeah, let's look at what we should be measuring. Oh wait, I just realized I don't have something for you to measure. But basically, this is the first step, right? I have my notes here that shows the typed out working. Basically, take this calibrated value, 1 EPG equals 2 micrometer. Use the eyepiece graticule, which is this, to measure a cell, and then figure out how many micrometers that cell is, what is the length is of the structure that you're measuring. That's it. All right. Now let's look at magnification instead now. Now in magnification, here's the compulsory definition that I have to give you. Please memorize this. Sometimes it comes out. Magnification is the number of times an image is enlarged compared to actual size. So obviously the formula would be image over actual. So how many times the image is enlarged compared with the actual size of the object. My students usually memorize this as MIA, which is missing in action. Remembering that is I over A. Now, there are 3 steps when it comes to this kind of questions. I always encourage students to convert first because the top and bottom should have the same units for this to work. Okay, and number 2, substitute in your values. What is I, what is A, and then calculate using the formula. Now, very often they would probably also ask you to derive the image size from the magnification and actual from the magnification and image, vice versa. Now, from this imagination of a little triangle, I use this to think to myself, okay, M equals to I over A, M equals to I on top and A below, right? And then remember that A, which is on the side here, equals to I over M, I on top and M below. And we can also see that I equals to M times A because they are on the same level here. So this is how I remember it. But you can use any method you want or just derive it from this formula on the spot. But this is the important bit. Remember to convert all measurements to the same units before you do any calculation. So for example, here's an exercise. A mitochondrion measures 36 mm in length. This is a electromicrograph, so it is a picture. It is 36 mm. 36 mm is 3.6 cm. That's huge. There's no way a mitochondria is 36 mm in real life, right? This is just the image size, not the actual mitochondrion size. This is the magnification. So what is the actual length of this organelle in mm? Pause the video, pause it now, and try it. Remember that your major formula is M equals to I over A. Have you tried it? Have you tried it? They want actual, which means you need to divide image by magnification. Right? They want it in micrometer, but they gave you millimeters. So how much is it? Try it, try it, try it. So if you tried it, the answer would be 36 over 30,000. However, this would result in an answer in mm, right? So to convert this to micrometer, you would want to multiply the answer by 1,000. And the answer would be 1.2 micrometer. Yeah, let's try another question, shall we? Let's try this one. So, this is a micrograph taken by electron microscope of an egg of an Aedes mosquito. If you don't know what Aedes is a species of mosquito that can cause a disease called dengue. It is very prevalent in tropical regions like Malaysia, where I'm from. But anyways, What is the magnification of image? How can we figure this out? Pause this video to think about it a second. The formula is the same. M equals to I over A. They didn't give you much information, but the information, pro tip, is in the image. Try it. Pause the video now. If you paused the video and tried it, here's the answer. So the only piece of information they give you is actually the scale bar right here. This scale bar is something that you see in maps as well. It's telling you that, okay, this length on the diagram is equivalent to 200 nanometers. So basically this here is actual 200 nanometers. What is the image size, right? The image size is the literal length of this line. So in front of my computer, I can reach up here and take a ruler, measure on my screen on the scale bar. I know you can't see me use it, but on my screen it says that it's 42 mm. If you remember, we always measure in mm, right? 42 mm. And yeah, voila, we can do our conversions. Okay, remember, convert it to correct units first. So I would convert mm to micrometer and to nanometer. So that is times 1000 and 1000 again. So it's 10 to the power of 10 to the power of 6. We should know how to do this by now. And then we divide them. So m equals to 42 times 10 to the power 6 divided by 200. And after I type the calculator, I would get, I just typed the calculator, I would get 2 times 210,000, 210,000 times. That's the magnification of this image on my screen. So if you're looking at this at a smaller like screen or bigger screen, you might get a different answer, and that's okay. Right? As long as you convert it right, you measure it right, you should get it right. Okay, this is the actual magnification of this image on your own screen. How cool is that? So yeah, just a few notes, by the way. When we talk about magnification, when we express the magnification, right, make sure that it's in whole number. That is a Cambridge requirement, and make sure there is a times sign in front, showing that this image is enlarged 210,000 times. So yeah, make sure it's in that formatting. Again, don't forget to convert before you do anything, just to make sure your answer is right. So yeah, these are the types of questions that you may encounter in your past papers, and I hope you learned something today. I'll see you next video. Bye bye.